Hello students, today we are going to discussing about the continuation of human reproductive system. Already we studied about the last episode, the last class under that is fertilization. What is meant by fertilization? The fusion of male and female gamete. Normally in human beings, the fertilization takes place ampullary isthmus junction, which one can present in female reproductive tract and all. There, after fertilization, the implantation one can be takes place. That means exactly after fertilization, 7th day, the implantation normally one can takes place the wall of uterus wall, where one can be takes place and all. Then further continuation and all, before implantation, the particular cell division one can involve in that zygote and all. That is, we call as embryonic development. There is particular steps one can involvement now. Now we discuss about that particular embryonic development, that continuation of reproductive system, embryonic development. Then, due to continuation of embryonic development only, we called as that is implantation, everything it will be included and all. In this, what are the process which one can involve that embryonic development? In this embryonic development, one can take place, the first step one can involve into this one, cleavage. That means zygote, which one can undergoes mitotic cell division inside that particular fallopian tubules. Their first multiplication one can take place, but the first mitotic division one can involve after completion of fertilization after 24 hours, or exactly if you want 30 hours, the first cleavage division one can take place. Then second cleavage division mitotically one can take place 40 hours after fertilization. The third cleavage after third day after fertilization. So like that, which one can started the mitotic cell division, then one can increase the number of the cells and all. On that fourth day, the cells which one can present 8 to 16 cell stage, it will be present. The cells which one can present in that multiplication of that particular zygote and all, we call as blastomeres. These blastomeres, then it will be forms. It will be look like a mulberry like that structure and all. That's why we call as 8 to 16 cell stage in that cleavage particular stage and all. We call as marula stage and all. That means in zygote, then the first after completion of 1, 2, 3 particular mitotic cell division, it will be forms into the mulberry like that structure and all. That is we call as marula stage and all. Marula stage. That means on that fourth day and all. Then fifth to sixth day, then normally one can involve into the which one can multiply and formation of blastocyst and all. Blastocyst. Later, that means on that sixth day nothing but it will be present 64 cell stage. 64 cells. It will be present. Sixth day. Then, but all blastomeres which one can involve that mitotic division, such type of we call as holoblastis. All cells which one can involving under. Then these blastocysts which one can be present at the stage of blastocyst are ready to implantation at the time of cell division, wherever it will be present fallopian particular tubules under which gradually moving and enter into the uterus wall and all. That means exactly after 7th day after fertilization or averagely between 6 to 9 days, the implantation one can take place. Implantation means, so 100 plus cells, wherever it will be present in zygote, then implantation it will be takes place under. In this blastocyst and all, it will be present 100 plus cells, it is ready to implantation. Implantation. The wall of endometrium wall of uterus. After observing, after attachment endometrium wall of uterus, this particular blastocyst and all, then the particular morphological changes one can take place, the endometrium wall of uterus. Whatever the changes, then we call as that particular uterus we call as decidua. After implantation, that uterus, that endometrium wall we called as decidua after implantation. According to their position under decidua basalis, decidua capsularis, decidua parietalis, differently present. Decidua basalis, capsularis, as well as parietalis, which one can be present under. Then at the time of implantation, there are certain changes. Suppose zona pellucida, if it become thicker, unable to implantation of the embryo, which one can be degenerated. Gradually, which one can reduce the thickness of that particular zona pellucida, then only ready to implantation. For that implantation, which one can promote corpus luteum, which one can persistent corpus luteum. Already in menstrual cycle we said luteal phase, corpus luteum, one can secretes progesterone hormone and all. Then if fertilization may not be takes place, then corpus luteum becomes degenerate and converts into the corpus albicans, which one can undergo menstruation and all. But here corpus luteum, which one can be maintained and secretion of the progesterone. But corpus luteum may not be present here. Instead of corpus luteum, which one can promote, one can maintain the maximum percentage availability of progesterone, the completion of that particular, that growth and development of embryogenesis and all, that is we called as human chorionic gonadotropin. In this one, HCG is equivalent to that particular corpus luteum and all, which one can equivalent to or luteinizing hormone corpus luteum, which one can corpus luteum, which one can stimulate progesterone. Here corpus luteum one can absent. Instead of this one, that human chorionic gonadotropin, which one can secreted by the chorion, one among the embryonic layer, and one can maintain proper amount, proper percentage of progesterone throughout completion of that embryonic development. So after implantation, one can stop the menstrual cycle. That particular stoppage, the period menstrual cycle, that condition we called as pregnancy. Whenever stop after implantation, one can stop regarding related to that menstrual cycle and all, that condition we called as pregnancy. That particular growth and development of embryonic period, scientifically we called as gestation period. In human beings, 9 months plus or minus 7 days it will be present. In pregnancy, that gestation period 9 months plus or minus 7 days it will be present in humans and all. But the maximum gestation period normally one can observable in mammals into the elephant, 624 days. The gestation period of in elephants, it will be present 24 days. Related to cat as well as dog, it will be present 60 plus days. Exactly if you want 63 days and all. Very least gestation period present into the rat. Within the month, one can complete that embryonic development, ready to deliver their young ones and all. That is, in this one, further development, which one can continuously inside that particular womb or metra or hystera of the uterus and all. Further development, one can be takes place and all. Then, what is the further development? That is, one can formation of this one, gastrulation. That is, gastrulation. Then what is meant by gastrulation? The blastocyst. In that blastocyst, already one can study about in that blastocyst nothing but once again one can recall the 64 it will present the outer layer, the inner cell mass. Both the two layers are present in this one. This is trophoblast, this is inner cell mass under. In that blastocyst, it will be present outer trophoblast, inner cell mass. This is the middle blastocele, it will be present under. This particular stage we called as that particular blastocyst. The cells, wherever the trophoblast, where one can attach the wall of uterus, we called as cells of Robber. This is we called as the cells of Robber. The cells of, why? Because inner cell mass further it will be equivalent to the embryo. It is equivalent to the embryo. This cell mass, it is equivalent to the stem cells ready to organize, ready to development, either tissues, either organ systems, any one among these one. Why? Because it acts as this one, stem cells. It is the further development of this particular one. For growth and development of that inner cell mass, further it is the embryo who will be provide that particular food material. That is the outer layer, trophoblast, outer layer, the trophoblast it will be present. Trophos means one can provide nutrients to the inner cell mass, the further multiplication, growth and development of embryo and all. Embryo in that sense, up to the first, after completion of the implantation, up to 2 months of that pregnancy of the mother, we called as embryonic development, that development of that particular zygote, we called as embryo. From 3rd month to the completion of that gestation period, we called as fetal development. That is the fetal development. Then related to gastrulation, blastocyst. So transformation of blastocyst into gastrula while formation of primary germinal layer with morphogenetical changes and all. That is we called as gastrulation. That means blastocyst converts into gastrula. The transformation of blastocyst, it converts into that gastrula with the help of primary germinal layers like mesoderm, ectoderm, endoderm with morphogenetical changes of the cells, rearrangement of the cells, which one can be present. That is we called as gastrulation. For this gastrulation, what are the steps which one can involve? How one can be changes that blastocyst to the gastrula stage and all. That is, in this first one, that embryonic disc, the formation of embryonic disc, that is the first one, that formation of embryonic disc. So here in exam point of view up to now, what is meant by morula, in what particular cell stage, at how many days after fertilization that particular implantation one can be takes place, which particular hormone one can maintain progesterone until completion of pregnancy time period of that particular mother, that is that HCG, which one can promote, which one can stimulate the progesterone continuation to maintain of this one. Then this formation of embryonic disc and all. This formation of embryonic disc already inner cell mass it will be present and all. Outer layer trophoblast cell, inner mass of cells it will be present. This is the trophoblast and all. This inner cell mass, this is the trophoblast layer. This inner cell mass, which one can differentiate two types of the cells and all. It will be differentiated two types of cells. The cuboidal hypoblast cell. This is hypoblast. Then that elongated columnar epiblast cell. That columnar epiblast. This is the actual trophoblast as well as blastocele, it will be present. So the inner cell mass, which one can differentiate the lower hypodermal, that is we call as hypoblast, it is the primitive endodermal layer. This is epiblast, that is important exam point of view. Which particular these two layers one can consider primitive endodermal layer. Hypoblast we call as primitive endoderm. Epiblast we call as primitive ectoderm under. Primitive, that is ectoderm, this is the primitive endodermal layer under. So these hypo and epiblast, that condition we call as embryonic disc under. This formation, these two, then one can consider embryonic disc under. Then after formation of embryonic disc, then next stage amniotic cavity. Then the formation of amniotic cavity under. Formation of amniotic cavity. How one can be formed this amniotic cavity, where it will be present? That is important. Amniotic cavity present between epiblast as well as trophoblast. It will be present in between epiblast as well as trophoblast. These particular layers, amniotic cavity, it will be present and all. This is the trophoblast. This is epiblast, columnar. Then apart from this one, that is the hypoblast cells, which one can be present here. In between that gap, one more layer. In between that gap, one more layer it will be present. This is amniotic cells. Amniotic cells, amniogenetic cells. These amniogenetic cells, which one can derive? That is important. From where amniogenetic cells one can be formed? Amniogenetic cells one can be formed from trophoblast. It is derived from trophoblast. Trophoblast cell. From here it will be formed. In between the space we called as amniotic cavity. Like this. So where one can be present between amniotic cell as well as epiblast cell. Then epiblast cell, it will be present. Amniotic cavity filled with amniotic cavity, it must be filled with amniotic fluid and all. It will be filled amniotic fluid. So like that between epiblast as well as trophoblast, or from trophoblast what one can form amniotic cells and amniotic cavity, it will be filled with amniotic fluid that is present. That is the formation of amniotic cavity and all. Then another very important one, formation of extraembryonic coelom. It will be formed extraembryonic coelom. How it will be formed? Where it will be arranged in that particular aspects and all. That is in third one, that extraembryonic coelom. Extraembryonic coelom. This extraembryonic coelom, while formation of extraembryonic mesoderm, then one can form and all. But extraembryonic mesoderm, there is no differentiation, there is no further formation of any organ, any tissue and all. This extraembryonic coelom, it will be formed in that trophoblast. Trophoblast will be present. Then these are columnar epiblast cells. It will be present. Then cuboidal hypoblast cells, it will be present. Then these are amniotic cells. Amniotic cells are present. Which one can be present? That particular amniotic cells. Amniotic cells. In this one, in this lower particular hypoblast cell, ready to formation of endoderm, ready to formation of endodermal cells and all. it will be formed endodermal cells. It will be formed endodermal cell. Endodermal cell, this is, that is extra embryonic, that is mesoderm. Whenever formation of extra embryonic mesoderm, then here it will be present, we called as yolk sac. This particular part we called as yolk sac and all. This is amniotic cavity and all. Remaining parts as usual it will be present. Then this, while formation of mesoderm and all, then it will be formed extra embryonic coelom. Then after formation this one, then coelom it will be formed and all. It will be developed that particular coelom. In that coelom, which one can reduce it. There is a primary yolk sac, it is the secondary yolk sac. Secondary yolk sac it will be present under. Here, which one can be present? The outer that mesodermal layer, one can form the outer, that it will be formed into the somatopleuric. It will be formed outer somatopleuric mesodermal layer. Somatopleuric, extra embryonic somatopleuric mesodermal layer. Inner splanchnopluric mesodermal layer it will be formed. This is the inner one, splanchnopluric mesodermal layer. This is the outer one, that is the somatopleuric, that is mesodermal layer, mesoderm. This is inner one, splanchnopluric mesodermal layer. In between the space, this is that extra embryonic coelom. Then it will be formed extra embryonic coelom, it will be formed under. So like that, this extra embryonic coelom, which one can be developed while formation of mesodermal layer and all, there is no differentiation of this one, which one can be present. Then after formation of extra embryonic coelom and all, here very important thing and all, the next step, that is how one can formation of chorion as well as amnion. How one can formation of chorion layer, how one can formation of amnion layer. Chorion layer, this is, in between these one can form into the chorion. In between these one, we called as amnion. In between this one, that is the yolk sac layer only. In this diagrammatically, this is the amniotic cavity, this is the yolk sac under, the outer one trophoblast layer it will be present. This is the trophoblast, which one can be present. This is the endodermal layer, Then the chorion, how one can be formed the chorion? Chorion formed the outer trophoblast, the inner splanchno, the inner somatopleuric mesoderm. Chorion one can form like a trophoblast. So that is important. Where it will be present, either outer or inner. Outer trophoblast. Trophoblast one can be present outer. The inner somatopleuric mesodermal layer, extraembryonic mesodermal layer. That is the somatopleuric. That somatopleuric mesodermal layer. This is mesoderm. So indirectly, in future, the trophoblast, which one can form into the that endo, there is ecto and all, this is the meso and all. This is we call as chorion. The future, the chorion, one can convert it as this one can formation of this one, placenta, which act as an important role to give nourishment from baby to the mother, from mother to the baby, which one can exchanging of gases as well as hormones, everything and all. That is important. Which part one can future, one can formation of placenta and all. From where one can originate that our answer will be the chorion. Then amnion. How amnion one can be formed? In this amnion, the outer, it will be present splanchnopluric mesoderm. This is splanchno, that is pluric mesoderm. Extraembryonic mesoderm, that is completely outer. That is the splanchnopluric extraembryonic mesoderm. Then this is the outer one. Inner, it will be present. Inner, which one can present regarding related to amniotic cavity. That amniotic cell membrane. Amniotic cell membrane, that is inner one. This is outer one. Like that. So, chorion and amnion act as a crucial vital role in human beings, which one can further growth and development of that particular fetal development in human beings and all. Then apart from this one, it will be formed, how one can be formed into the germinal layers. After formation of this one, then ready to formation this one, germinal layers and all. What is the germinal layers? Normally it will be present, 3 germinal layers are present. Then fourth one, the formation of that germinal layers. The formation of germinal layers. In this particular area, how the germinal layers are formed. In this one, only one can observe this particular part only, one can extend it. In this one can form into the first primitive streak. First one can form into the primitive streak. Then only it will be developed. The 3 germinal layers one can be present. That is the ecto, meso, endo. But exam point of view, one can remember what is the first germinal layer one can be formed in human, that growth and development of embryonic development and all. Then your answer will be the endoderm. They will give endoderm, mesoderm, that is ectoderm and all. Then your answer will be the endoderm, first germinal layer. The first germinal layer, that is endoderm, it will be formed. After formation of primitive streak normally one can form into the epiblast. There it will be formed. In that one, invagination of number of cells inside this one. After invagination of these cells, then some of these cells, which one can be proliferate into the hypodermal one, then formation of endodermis and all. Then that particular mesoderm, like this it will be formed and all. But this is very important and all. In this, for observing rough diagram and all, in this one, this is epiblast cell. That one among this one, epiblast cell. The epiblast cell, this is we called as that primitive streak, the epiblast cell. Then that below already hypoblast cells it will be present. That is hypoblast, which one can be present. In this, as usual, it will be present. Then it will be invagination of number of cells, invagination of the cells. That is invagination of cells between epiblast as well as hypoblast, invagination of cells. Some of these invaginative cells, which one can proliferate or adjust, which one can insert towards that hypoblast, and formation of newly formation of endoderm and all. So from hypoblast, very important for formation of endodermis. Endodermis and all. Then after invagination, it will be formed normally. It will be formed as usual and all. It will be formed normal epiblast, then hypoblast, then middle of this one, middle of this particular group of the layers we call as, this is the epiblast, this is the epidermis, this is the mesodermis, this is the endodermis. So after primitive streak, a group of cells are formed. After cells are formed, it will be invaginate, it will be, which one can be adjusted towards that hypodermal one, then it will be converted as this one, newly formation of that endodermis under, then first endodermis one can be formed, then epidermis, then mesodermis, like that, three germinal layers, which one can frequently one can be formed. Then apart from this one, then extra embryonic layers also it will be formed. That embryonic membranes. These are the main normal membranes and all. Extraembryonic, what is the extraembryonic membrane? There is a chorion, there is the amnion. Already we discussed here. There is a chorion as well as amnion. Both are acting as a very important role. Along with this chorion and amnion, it is also present that allantois as well as egg sac. It will be formed. That is the egg sac as well as allantois, that chorion, that is amnion. These two act as in exam point of view, how many extra embryonic membranes are formed in human being, then your answer will be the all four. But among these four, which is act as a very important role, that is the chorion and amnion. Even primitively one can form the egg sac and amnion. Egg sac, which one can form from the gut, that there is actually allantois. It will be formed the gut of that embryo. It will be helpful only that particular which one can purification of blood vessels in that umbilical cord. Primitively. More than that one, there is no particular function at all. Then egg sac, in future, it acts as an important role for formation of that particular blood cells in primitive stage, embryonic stage, formation of blood cells with the help of egg sac. More than that, it is not at all any further function. That chorion acts as important role further, one can formation of complete placenta. That amnion, which one can protective one, which one can shock absorber, which one can prevent electrical shocks, one can protection until completion of that particular gestation period and all. In this egg sac, here, if you observe very rough diagram, diagrammatically if you observe here, that is, which one can be present, that is trophoblast, it will be present. Among these one, it will, that embryo, this is the embryo. In this embryo, this is the posterior end, this is the anterior end. In this one, in these particular aspects, which one can, it will be covered with the amniotic cavity. This is the amniotic cavity, it will be covered. This is embryo, there is a mass of cells under. In this gut wall, it will be present, which one can originate it, that is the one among this egg sac, from this gut wall it will be present allantois. This is the egg sac, this is allantois, it will be formed. Apart from this one, that which one can somatopleuric, splanchnopluric, that is a somatopleuric, which one can be present all along with this particular layer. Somato, which one can be covered up to here. Up to here it will be present and all. Then it will be covered all along with these egg sacs and every aspects and all. It will be aspects. Like this, allantois How allele types will be present? The outer it will be present mesoderm. Inner it will be present, that is endoderm. Endoderm it will be present. Here also, yak sac also, which one can be present? Outer, that is related to the meso, that is one among the somato, that is a pleuric mesodermal layer. Then inner, which one can be present? Related to endodermal layers and all. But here trophoblast, that is ecto, that is regarding related to somatopleuric. But here splanchnic, that amniotic, this already we discussed and all. So here another very important, from this particular layer, which one can be formed? Chorionic, that particular chorionic villi, which one can be developed? That chorionic villi and all. That is chorionic villi. The further which one can made it, which one can pit, which one can penetrate the wall of endometrium of this uterus, for which one can absorb for food material, which one can absorbing for further growth and development of that particular embryonic development. So chorionic villi, which one can penetrate towards the endometrium wall of uterus. Further it will be developed as this one placenta. According to formation of placenta, choria, that's why it is called as chorionic placenta. Then it is bathed with the blood vessels, that's why hemochorial placenta. Then this placenta, it will be present, which one can divertly. That is we called as metadiscoidal. That's why different types of, according to its position, hemochorial, metadiscoidal, as well as chorionic. That three different types of that placenta, which one can present. It acts as a very crucial role. One can transmit, one can distribute the nutrients from mother to the fetus, as well as similarly exchanging of gases. Suppose carbon dioxide from fetus to mother, nitrogenous substances from fetus to mother. From mother only, which one can excreted and all. Apart from this one, it is teratogens. Some of these, any harmful, dangerous one, like alkaloids, like any drugs, like any viruses, which one can enter. There is a multiple disorder of embryonic development one can be takes place. One among these, that particular chemical drug, that is the thalidomide, that is it, which one can, that is the thalidomide. It is one type of this teratogen, one type of this harmful one. It may be causes multiple disorder of this one. So towards that placenta, so placenta act as that much important. Apart from this one, this placenta one can store the food material. That is in the form of glycogen. It will be stored, that particular, that placenta. And apart from this one, it acts as a hormonal action. It will be secreted different types of the hormones. What type of hormones one can secrete the placenta? It will be secreted HCG as well as HCS. It is also that chorionic, that thyrotropin, chorionic, that thyrocorticotropic, as well as relaxin, as well as estrogen, as well as progesterone. Like that, multiply, these hormones also one can be secreted and all. Multiple, that is a chorionic thyrotropin, chorionic thyrocorticotropin, relaxin, all are, so that human chorionic gonadotropin one can maintain progesterone. Then human chorionic somatomammotropin one can growth and development of mammary glands at the time of that pregnancy and all. Then remaining all one can supporting that normal proper development of that particular things and all. Relaxin one can helpful at the time of parturition condition.